Hello, everyone. Thank you for joining us today for the 10th webinar in our series on the use of NAMs and risk assessment. This series is being co-organized by the Environmental Protection Agency, the Physicians Committee for Responsible Medicine, and the PETA Science Consortium International. I'm Bridget Rogers. I'm an advisor to the PETA Science Consortium International, and I'll be co-moderating today's webinar with Gino Scarano, who is a senior advisor in the New Chemicals Division of the EPA Office of Pollution Prevention and Toxics, as well as Christy Sullivan, who is the Vice President of Research Policy at the Physicians Committee for Responsible Medicine. We have two speakers today who will be presenting on data-driven solutions to reducing animal use and ecotoxicity. First, we'll hear from Michael Lowit from the EPA Office of Pesticide Programs, and then we will hear from Carly Lalone from the EPA Office of Research and Development. Before I introduce our first speaker, I'd like to point out that the slides and a recording from today's webinar will be posted online at the link on the screen shortly after this webinar. Slides and a recording of the first 9 webinars in this series are also available at this link. There will be time for questions for both speakers at the end of the second presentation. Everyone is on mute, but you can type any questions into the questions section within your GoToWebinar toolbar, which should be on the right-hand side of your screen. Please indicate your question if it's directed to a specific speaker. I'll now turn it over to Gino Scarano for his introductory comments. Gino?
Thank you, Bridget, and welcome all. As Bridget said, this is the 10th one, so I feel this is an opportunity to reflect. We have 10, so that's a substantial number. So this wonderful series, which I'm really happy that PCRM and PETA have handled all of the logistics and have done an amazing job. And so when you look back at the 10, this will be the second one where we focus on on environmental organisms and environmental tests. So I think that's important. And the big winner for the most number of webinars is sensitization, which is no big surprise. And we had a good mix of different tools and at least one TSCA-specific discussion of NAM nominations, which is something that's near and dear to perhaps most people's hearts as we've been operating under Lautenberg and the new Section 4H for 5 years now, almost 6. So I just want to say that. And the second thing before we really get to the important part of this, which is obviously to hear Michael and Carly, is I think most of you may be aware of the collaborative research program that we have begun talking about with the Office of Research and Development and our group, the New Chemicals Division in OPPT. And a big part of that is, I think, going to be bringing NAMs into the New Chemicals program, which many people have been talking about for a long time. And we had a public meeting 3 weeks ago where we had this 5 research areas identified to bringing both, updating our process and innovating the science and how we do our work, and NAMs play a central role in that. So I urge people to look at our website, just look at Collaborative Research Program, and you should be able to find it and take a look at that. With that, those are the introductory remarks I wanted to say, and I'll turn it back to you, Bridget, to introduce our speakers and hear the content. Thank you.
Great, thanks so much, Gino. Okay, speaking first today will be Dr. Michael Lowit. Michael is a senior scientist at the EPA Office of Pesticide Programs in the Environmental Fate and Effects Division. He has worked there for over 12 years, conducting ecological risk assessments in support of regulatory decisions on the registration of conventional pesticides. He is involved in OPP's efforts with new approach methodologies and currently represents OPP on the Ecotoxicity work group of the Interagency Coordinating Committee on the Validation of Alternative Methods and the Validation Management Group for Ecotoxicity Testing of the Organization for Economic Cooperation and Development Test Guidelines Program. Welcome, Michael. Please go ahead.
Thank you. Can you all see my screen slides?
Yes, we can. Right now, it's not in presentation mode.
Okay. There we go.
Great, looks good.
Okay. Good morning, everyone. Thank you to PEP for the opportunity to participate in this webinar. It is these types of events that set the stage for dialogue and building collaboration on advancing the science and incorporating NAMs into regulatory frameworks. As Bridget said, I'm Michael Lowit from the Environmental Fate and Effects Division, or EFED, of the Office of Pesticide Programs, OPP, at the US Environmental Protection Agency. My presentation is a high-level overview of EPA's perspective on applying the 3 Rs to ecological risk assessment in support of the registration of pesticides. We'll be focusing on ecological risk, but other divisions such as the Health Effects Division, or HED, have their own efforts. So first, just a little review. OPP has a long-standing commitment to the 3 Rs principles, which are to reduce animal use, for example, fewer whole animal studies, replace laboratory whole animal studies by the implementation of in vitro studies, which are generally meant to indicate studies with cells and tissues, in chemical studies, which are abiotic chemical reactivity assays, in other words, no cells are used in those, and computational approaches, also called in silico, which include methods like the use of the quantitative structure-activity relationships, or QSARs, to predict toxicity. And finally, refined study protocols to reduce suffering. So for example, this might be the modification of an existing study design to lessen or avoid animal pain and distress, or it could simply mean requiring fewer individuals in the existing study design. So an important question to ask is, why would we want to apply the 3 Rs principles to the way we currently assess risk from pesticide use? First is for reasons of ethics and animal welfare. Do we need whole animal testing to be protective of human health and the environment? When is it necessary, and when maybe less so? The second reason is efficiency. Theoretically, there should be savings in both time and money once 3 R strategies are adopted. The third reason is public health. This gets at the idea that it's known in some cases that test organisms aren't the best models for humans, and that in vitro studies with human tissues and associated modeling have greater human relevance and can lead to improved science. A parallel idea for protecting the environment might be using in vitro studies to test a greater number of taxonomic groups, rather than simply relying on in vivo testing with one taxonomic group as a surrogate for other taxonomic groups. And a fourth reason is expectations, which I'll address in the following slides. What does OPP do, and why are we concerned with animal testing? OPP is charged with regulating the use of all pesticides in the United States. The standard approach is to base ecological risk assessments on toxicity data primarily derived from whole animal studies, which are required in the Code of Federal Regulations, or 40 CFR Part 158, for the registration of conventional pesticides. It's important to recognize that the generation of that data can be costly and uses many individual animals for the registration of a single pesticide. Costs can exceed $100 million, and the number of animals tested can be in the tens of thousands. It's critical that we work with outside partners to advance the application of the 3 Rs in a regulatory context. To that end, we are working with a wide variety of stakeholders to systematically look at the studies we require for registration, looking for opportunities to provide waivers to reduce testing and costs without negatively impacting our ability to protect human health and the environment. We're also active in work to advance the use of new approach methods, or NAMs, in regulatory risk assessment, although those activities generally have a longer time horizon for regulatory acceptance. As part of advancing these efforts, OPP staff are active participants on various committees of ICBAM, which is the Interagency Coordinating Committee on the Validation of Alternative Methods.
Michael, I'm
Yes.
so sorry to jump. I'm so sorry to jump in, but your sound is going in and out for some reason. Are you using a microphone?
Just the normal computer. Can you hear me now?
Yes, maybe just try to make sure you're not turning your head or anything, that you're kind of pointing your voice right at the mic.
Yeah, I'm not even sure where it is on the computer, but.
I know you sound good right now. I'll let you know if it happens again.
Okay, great, thanks.
Sorry. Samantha.
No, thanks for letting me know. Is the volume okay though? It's just the in and out. Okay. Yeah. Okay. ICFAM is an important stakeholder group consisting of representatives of 16 federal regulatory and research agencies that require, use, generate, or disseminate toxicological and safety testing information. ICFAM facilitates the development, validation, and regulatory acceptance of test methods that replace, reduce, or refine the use of animals in testing. My science division in OPP, the Environmental Fate and Effects Division, is an active participant on the ecotoxicology work group of ICFAM. The real driving force of ICFAM is NICETA, which is the National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods, which is part of the National Institute of Environmental Health Sciences, and they provide scientific and operational support for ICFAM technical evaluations and related activities. The data requirements in Part 158 provide the scientific basis for characterizing the potential risks associated with pesticide exposure. However, there is flexibility in implementing Part 158, and OPP has a long history of practicing that flexibility. Additional data can be required, alternative approaches can be accepted, and studies can be waived. In 2013, guidance was released by OPP on guiding principles for data needs. The purpose is to provide consistency in the identification of data needs, promote and optimize the full use of existing knowledge, and focus on the critical data needed for risk assessment. The guiding principles re-emphasize the flexibility in implementing data requirements in the context of new and emerging tools, which may be used to support risk assessment and risk management decisions. In 2016, the director of OPP released a letter to stakeholders reinforcing OPP's commitment to the 3 Rs and highlighting 3 main objectives, which are to critically evaluate the studies that impact OPP decisions on pesticide registrations, expand the acceptance of alternative methods, and reduce barriers with stakeholders and other regulatory agencies in the advancement of adopting alternative methods. Fast forward to 2019, the US EPA put out a directive from the administrator prioritizing efforts to reduce animal testing. One of the priorities of these efforts is to host an annual conference on the state of the science on the development and use of NAMs for chemical safety testing. As defined in the work plan, new approach methodologies refer to any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment to avoid the use of animal testing. The final reports for the first 2 conferences are available on the website listed. The 3rd conference is planned for later this year. Another priority in the directive was the development of a work plan focused on the development, testing, and application of methods that allow for the reduction in whole animal testing while remaining protective of human health and the environment. EPA's NAMs work plan was created to prioritize agency efforts and resources towards activities that aim to reduce the use of animal testing. This work plan was developed by experts across the agency to set the objectives and strategies for using NAMs. The work plan was updated in December 2021 with the main objectives and strategies being unmodified. The NAMs work plan identifies 5 objectives for achieving the reduction goals while ensuring that the agency's regulatory compliance and enforcement activities, including chemical and pesticide approvals and agency research, remain fully protective of human health and the environment. One of the useful aspects of the document is that it lays out strategy, deliverables, and timelines for accomplishing the objectives. However, like any roadmap, the work plan represents a snapshot in time, and this document will need to continue to evolve as EPA's knowledge and experience grows. The agency is committed to regularly reviewing the work plan to ensure that the objectives, strategies, and deliverables provide the best possible path to success. So now I'll quickly go through the 5 objectives. The first of them is to evaluate regulatory flexibility for accommodating NAMs, which seeks to determine how much regulatory flexibility exists for each EPA program requiring toxicity studies and any roadblocks that those programs may have applying NAMs for regulatory purposes. As mentioned earlier, OPP has previously identified that it has flexibility in implementing Part 158 data requirements through waivers and alternative approaches. The second objective is to develop baselines and metrics for assessing progress. This is to determine how many animals are used in a program and track how various efforts have reduced that number. The third objective is to establish scientific confidence in NAMs and demonstrate application to regulatory decisions. Scientific confidence associated with in vivo tests comes from the decades of experience in their development and application. As we transition to the use of NAMs for making regulatory decisions, we need to continually build more scientific confidence in the information from those approaches, while also establishing the appropriate expectations for their performance and demonstrating their application to regulatory decisions. In part, this means that regulators work with method developers to ensure that NAMs, assays, and approaches are fit for purpose and have a domain of applicability that is suitable to address the data needs of a specific regulatory context. The fourth objective is to develop NAMs to address scientific challenges and fill important information gaps. In other words, what NAMs do we need to alleviate the need for whole animal testing? The data gaps that need to be filled or the scientific challenge to be addressed is tied to the specific regulatory needs to be addressed. Therefore, a single NAM or combination of NAMs will unlikely be applicable in all need contexts. And the final objective is to engage and communicate with stakeholders. I can't stress this one enough. Widespread use and reliance on NAMs in agency decisions requires a fundamental change in thinking, given that traditional approaches have been used for decades. Given this, effective engagement and communication with stakeholders is essential to increase acceptance, obtain constructive feedback, and improve the acceptance of using NAMs to inform agency decisions. So there are a variety of strategic plans and roadmaps out there. This slide shows one from ICFAM and 2 from EPA. They're generally similar, but really what I want to highlight here is that each one indicates that the introduction of NAMs into a regulatory context is an iterative process, which is heavily dependent on collaboration among stakeholders, specifically the assay or model developers and the end user, such as the regulator. Early and continued engagement with the model or assay developers increases the likelihood that a particular NAM can be used in a particular regulatory context. In other words, is it appropriate and fit for purpose for making regulatory decisions? So the remainder of the presentation, I'm going to focus on one retrospective project in the Environmental Fate and Effects Division of OPP that may lead to reductions in animal testing. Retrospective, I mean that we're going back through our existing data sets of laboratory studies to determine which studies have the greatest impact on risk assessment conclusions and which ones maybe didn't have as much impact. Although I will focus on one current retrospective effort, we have many ongoing projects, as well as some that have been completed and have led to guidance on potential waivers of specific studies. Naturally, there may be concerns that using fewer animals in testing could impact our mission to protect human health and the environment. However, it's important to stress that while OPP is committed to reducing animal testing, it is not at the expense of protecting human health and the environment. We only will move forward with any of the 3 R's once we are satisfied that the alternative is ready for a policy change. So today I'm going to talk to you about our acute toxicity to fish retrospective. In collaboration with NICEDM, we're assessing the need for acute toxicity testing with fish for ecological risk assessments in support of registration decisions on pesticides. OPP ecological risk assessments use studies with cold freshwater, warm freshwater, and saltwater fish species to assess acute risk to fish. For a single chemical registration, this can result in acute toxicity testing with 200 or more fish. So our question here is if there is a consistently more sensitive fish across pesticides, and can we reduce data sets to 2 or even 1 fish study? Examples of considerations include patterns in chemical mode of action and interspecies variability. The analysis is focused on 181 conventional pesticides registered from 1998 through 2016. And this date period was selected to provide a sampling of the most recent chemical class, reasoning that these classes are the least mature in terms of the addition of new compounds within the class. In other words, this data set likely represents the classes new chemicals will likely be submitted for future registrations, and which decisions of fish testing will be required. The initial data set that we had to work with includes over 700 studies representing nearly 350 test substances, including active ingredients, their transformation products, and product formulations. The analysis is limited to test guideline studies, which is important for this comparison and reduces experimental variability as much as possible. The data set was curated by excluding studies that would not be acceptable for quantitative risk assessment. In other words, we excluded studies that we had lower confidence in the results. Bins were created with the remaining studies based on common test material. For example, only studies of the same formulation would be binned together. An analysis group was created for each test material tested with at least 1 cold freshwater, warm freshwater, and saltwater fish species, which are the 3 species required under current data requirements. So we've identified 110 analysis groups, some of which contain more than 3 species, but always 1 cold freshwater, warm freshwater, and saltwater fish species. And the plot here shows those analysis groups on the x-axis sorted by the mean toxicity of the test material, decreasing from left to right, with the LC50 values of individual studies comprising each analysis group on the y-axis. The general mode of action representation is relatively similar among fungicides and herbicides, with slightly fewer insecticides, in addition to 6 chemicals that didn't fall into one of the 3 major mode of action categories. Species representation of the tested species is reflective of EPA test guideline recommended species, with rainbow trout comprising 99% of the cold freshwater fish, bluegill sunfish comprising 64% of the warm freshwater fish, and sheepshead minnow comprising 95% of the saltwater fish species. The basic question is which of the tested species is most sensitive and can be determined for 60 of the analysis groups, which are termed non-equivalent. Those chemicals are roughly plotted within the dashed blue oval. A large subset of the analysis groups represents test materials that caused no mortality in any tested fish species at limit test concentrations or above the functional solubility limit of the test material. Therefore, the relative sensitivity of those fish could not be determined. That said, the toxicity of these compounds is considered functionally equivalent to the tested species from a risk assessment perspective. Analysis groups with greater than LC50 values above the limit concentration for all tested species are roughly shown in the solid yellow oval, and the limit concentration is 100 milligrams per liter. Generally, chemicals that are practically non-toxic or LC50 values greater than 100 milligrams per liter are classified as practically non-toxic and are not of risk concern, as exposure concentrations are typically at much lower concentrations than those tested in these studies. The remaining 10 analysis groups are termed unclear because at least one of the LC50 values is definitive, so an equal LC50 value. But something about the combination of the available LC50 values makes it uncertain which of the 3 species groups is most sensitive. The uncertainty in these cases is mostly attributed to test concentration selection and solubility issues, especially differential solubility in the freshwater versus the saltwater media. For example, one chemical has definitive LC50 values of 0.7 and 0.8 milligrams per liter for 2 of the species, but the 3rd species was tested at a lower test concentration and had an LC50 value of greater than 0.5 milligrams per liter. The tested freshwater fish were more sensitive than the tested saltwater fish in most of the analysis groups, with one of the tested cold freshwater species being the most sensitive at about half. There is no pattern in terms of general mode of action. Any potential patterns in chemical class haven't been determined at this time, in part due to the high number of chemical classes relative to the number of analyses. That said, there are a couple cases where there are at least hints that there might be some patterns within some chemical classes. In terms of hazard classification, freshwater fish tended to be more sensitive than saltwater fish to the most toxic chemicals in the data set. We continue to work on the analysis, and the results will be used to inform if there are situations where data waivers may be suitable for testing fewer species. In other words, what studies might be waived, under what conditions, which chemicals, and any assumptions that would need to be made in the absence of studies. In closing, OPP is committed to reducing animal testing burden without compromising the quality of risk assessments. This includes efforts by EFED to lead change in environmental risk assessment. Progress on incorporating reduced animal testing efforts into a regulatory context requires collaboration across many sectors, transparency, and peer review. If you have further questions, you can visit the ICFAM website or send an email to the all I've got. Thank you for your time and attention, and I hope the audio was okay the rest of the presentation.
Great, thank you so much, Michael. It was a great presentation. The audio was fine. As a reminder to everyone, we're going to hold all questions to the end, but please feel free to type them in as you have them on the GoToWebinar's questions panel, and we'll ask them after our next presentation. And again, please note if your question is for a specific speaker. Okay, speaking next will be Dr. Carly Wallone, a research bioinformatician at the Center for Computational Toxicology and Exposure within the EPA's Office of Research and Development. Carly has over 12 years of experience at the EPA and currently leads a research team that integrates computational, laboratory, and field-based studies to evaluate a variety of pharmaceuticals, pesticides, and chemicals for ecological risk assessment. She also serves as chair of the Society of Environmental Toxicology and Ecology North American Science Committee, as well as chair and co-founder of the International Consortium to Advance Cross-Species Extrapolation and Regulation. Carly, thanks for being here today. Please go ahead.
Well, thank you. Thank you to Amy and Bridget, as well as Christy and others for organizing. I'm happy to be able to come here and speak to you today, especially because it was so nice of Michael to set up this presentation very well, thinking about new approach methods, and particularly I'm going to talk about in silico new approach methods, particularly the sequence alignment to predict a cross-species susceptibility tool, and how we can utilize this tool to extrapolate knowledge computationally. So just as an overview, I'd like to talk to you about cross-species extrapolation and thinking about how bioinformatics can be used to advance extrapolation. I'm going to get into some details, but not too detailed, about the SECAPAS tool and some of the demonstrated applications of this methodology. Then I'd like to talk about how we're working to bring together champions in the field to advance the science for action through this international consortium to advance cross-species extrapolation in regulation. So we're understanding the effects of chemicals across species is very important for a number of different reasons. For the US EPA, we have a number of legislative mandates that we're interested in making sure that we can be protective of both human health and the environment. And to be able to understand toxicity of chemicals, historically, we've utilized whole organism testing, and more so, we're learning about new approach methods and the utility of high throughput screening assays, high throughput transcriptomics, and the utilization of the adverse outcome pathway framework that allows us to link molecular level changes, such as those that are evaluated through new approach methods, to apical level effects that were typically used for making decisions, and particularly risk assessment. So with these historical animal tests and the available data, as well as we're moving to new approach methods, we still need to be able to advance our methods in species extrapolation, really taking advantage of what we know about these model organisms, and utilizing that to understand their surrogates and how well they are surrogates for the diversity of species in the environment. So we know that we utilize model organisms, and typically in using model organisms, we have identified them because they're easy to maintain and they have good breeding capabilities or short lifespans, rapid life cycles. There's an ability to control their surroundings. And many times they require limited space and don't have as time-consuming care. And not always have they been selected due to, say, an understanding of their ability to represent other species within their taxonomic group or across species. And so that's why species extrapolation is quite important. And what is species extrapolation? So certainly it's using the knowledge about a species or a handful of species to be able to estimate, predict, or project effects or to other species. And in the context of chemical safety, that's dealing with toxicity and being able to project the trajectory of that toxicity to other species. And so why is this important? Well, we know that data availability for multiple species for chemicals is relatively limited. And so we also know that we can't generate new toxicity data, certainly not whole organism toxicity data, across the diversity of species. Further, we know testing resources are more and more limited, and we have an international interest to reduce and replace animal use in toxicity testing, with an ever-increasing demand to evaluate more chemicals in a timely manner, and sometimes this can be in an expedited manner. So we need to really take advantage of those scientific methods, scientifically sound methods, for understanding the sensitivity of a species to a chemical, and thinking more broadly about the diversity of species that could be exposed to chemicals in the environment. And this becomes quite an important challenge when we're thinking about threatened and endangered species, such as those that are covered under the Endangered Species Act. So where could we begin in understanding species similarities and differences using bioinformatics types approaches? So we can look at the existing and expanding databases that exist for sequence and structural information. So when I'm talking about sequence and structural information, I'm talking about proteins. There's large databases that curate the sequence information that's being generated. And there's large efforts, consortiums even, that are focusing on generating data and sequence information for whole genomes, for many different types of taxa, not just your common model organisms. And these large databases are really available for hundreds of millions of proteins representing hundreds of thousands of species. And so if we're thinking on understanding species similarities and differences, we can utilize the science of bioinformatics, which combines mathematics, information science, and biology to answer biological questions and really explore large volumes of data. And typically this pertains to DNA and amino acids involved in sequence of proteins. But what's really great about bioinformatics is that it's an approach that allows computers to do the work for us. So to utilize these approaches in bioinformatics for a particular purpose in thinking about species extrapolation, we need to begin relatively simple and advance as the science advances. So when we think about predictive toxicology and thinking about similarities at the molecular level across species, we can start doing sequence comparisons, understanding how similar, how well conserved a protein is, and then further move on to structural comparisons and certainly functional comparisons as well. So we can consider the sequence and structural attributes as a starting point to understand protein conservation across species. So this is important because chemicals tend to interact with proteins. And so what we wanted to do is develop a streamlined tool, very transparent for the data being used, to be able to extrapolate knowledge from one species to the diversity of species. And the tool that we developed is called the Sequence Alignment to Predict Across Species Susceptibility, or SEQAPAS for short. The tool version 1 was released in 2016. It's publicly accessible and the URL is provided there. And it really has been built specifically and in partnership with our program offices at EPA for both researchers and decision makers. So for utilizing the SEQAPAS tool for extrapolating knowledge across species, I wanted to talk a little bit about the required information for a SEQAPAS query. So first of all, it depends on what we know about a chemical protein interaction. So as far as, a pharmaceutical and in some cases pesticides, we may have knowledge of a targeted or a sensitive species. And so that species, the protein from that species would be the initial query information. In the context of utilization of high throughput screening knowledge, we may understand the model organisms used in that in vitro bioassay, and therefore that model organism could be used as the query species and the sequence for that particular in vitro assay. And then further, in the context of adverse outcome pathways, we may have developed AOPs and particularly molecular initiating events based on a handful of species or a single species. So those species for which the AOP had been developed and there's proteins involved in those key events, we can utilize those species to begin a SEQAPAS query. So once we understand how that chemical interacts with the protein in a particular species where we know they're either sensitive or that chemical has been developed to interact with that species for a specific purpose, we can take that chemical molecular target in that targeted species and compare it to those hundreds of millions of proteins from hundreds of thousands of species, and understand how well conserved that protein is in other species. So here we assume that the greater the similarity, the greater likelihood that that chemical could then interact with that protein in that other species, and therefore that conservation allows us to predict chemical susceptibility across species. Now, the SEQAPAS evaluation was developed to be very flexible based on available data. So there's 3 levels to this evaluation, with each level requiring a greater depth of knowledge for a chemical protein interaction. So at level 1, you could take any species, right? So we understand that perhaps it's it could be that we have mammalian data. It could be that the high throughput screening assay was developed with, rabbit. It could be that we're looking at a pesticide, so an insect would be of interest, or it was an herbicide, so we have a plant that's targeted with this particular chemical. And so in that context, we can take and compare that sensitive or that model organism at level 1 and compare the primary amino acid sequence of that protein, the full protein sequence, to all other species and understand how well conserved that full protein is across taxa. At level 2, if we have information about functional domains, such as a ligand binding domain, which the tool helps guide you to identify if there are known domains, we can compare those specific conserved regions across species. Then further, if we have a greater level of detail about that chemical protein interaction, and perhaps we know where hydrogen bonds occur with amino acids, or there's some information in the literature on site-directed mutagenesis, or say field resistance to pesticides that points out individual amino acids, we can compare those specific chemical protein interactions across species looking at critical amino acids or close contact amino acids. And what we're doing as we move through these levels of the CECAPAS evaluation is we're gathering lines of evidence towards protein conservation, and it actually adds taxonomic resolution to our susceptibility predictions. So what do I mean by that? I mean that at level 1, when we're comparing the full protein sequence, we might be able to make predictions between vertebrates, invertebrates, plants, etc. But when we get to level 3, where we're looking at those critical individual amino acids and chemical specific interactions, we can make predictions at the species level. So the prediction from CECAPAS, we give a yes or a no answer. So basically, we're saying yes, there's enough information that's available for demonstrating conservation of that structure that we would predict likely susceptible or likely conserved, or no, there's not enough information, not enough similarity between the sequences where we would say that they're conserved. And so really there's a lot of data that goes behind making that prediction, but the ultimate prediction is a yes or a no, susceptible or not, conserved or not, across hundreds or thousands of species. And so one of the things that I think is important to point out is that CECAPAS does not predict the degree of sensitivity or susceptibility. So if we think about the factors that make a species sensitive to a chemical, there's many different factors, including toxicokinetics and life stage, life history. But again, that very specific question we're asking is that known target for the chemical, is it available or not? Some of the strengths of CECAPAS, it's publicly available to everyone. We have multiple lines of evidence that we can pull together pretty rapidly for conservation for hundreds of thousands of species and make predictions very rapidly. It takes advantage of well-established tools and databases and it doesn't require somebody to have a very strong understanding or to have been working with these tools for a very long time to be able to utilize the CECAPAS tool, because it's very streamlined, consistent, transparent, and it has multiple published publications to support the methods, as well as whenever we develop different features of the tool that have been requested, say from some of our program office partners or through some of the research we've done, we always generate case examples to demonstrate the application of the tool for that particular purpose. And typically this is in line with needs from our decision makers. And it also guides users to appropriate input. So we wanted this tool to be available to people that have a basic biology background, not necessarily a strong background in bioinformatics. And then certainly one of the things that we always keep an eye on is the evolution of the field of bioinformatics approaches and being able to make the tool more user-friendly through smart automation or semi-automation. So keeping that in mind, I wanted to just chat briefly on some of the applications of the CECAPAS tool. So we've utilized the tool to develop a number of different case studies, and we've published a number of these. Most recently, that we just submitted a paper last week, was on the extrapolation of adverse outcome pathway knowledge across species. So this is in the context of defining the taxonomic domain of applicability for an adverse outcome pathway. When we have information or an AOP was developed for a particular species, in this case, Apis mellifera or honeybee, what could it tell us and how applicable is it to other bee species, including non-Apis bee species? And then we've utilized a tool to extrapolate high throughput screening data. So in the context of estrogen receptor alpha, androgen receptor, steroidogenic enzymes, and the thyroid axis, we've utilized the tool to understand how broadly we can anticipate extrapolating high throughput screening data such as that generated from ToxCast. And we've published on that as well. And there's hundreds of ToxCast assays that all represent gene targets with model organisms associated with those assays. And so we did do extrapolation from those ToxCast assays to the diversity of species and have made that data publicly accessible through the CompTox chemicals dashboard, as well as through SequPass itself. We've used the tool to predict relative intrinsic susceptibility. So this has been in the context of multiple pesticides, thinking about the Endangered Species Act, as well as in thinking about how we can expedite derivation of aquatic life criteria. We've used the tool to predict bioaccumulation potential across species, and this has been in the context of chemicals of concern such as perfluoroalkyl substances, and we've published on that. We can utilize the tool not just for extrapolation, but also for generating research hypotheses, and we've demonstrated that in publications thinking about strobilurin fungicides, as well as in prioritization strategies. And so because I'm not able to get into the examples in great detail for all of these, I just chose one on pesticides to follow up Michael's talk, and particularly thinking about the use of SequPass for butterflies and the evaluation to predict chemical susceptibility to threatened and endangered species. So we know that butterflies are in the order Lepidoptera, and we understand that there are insecticides that are molt accelerating. And typically these molt accelerating pesticides target the ecdysteroid receptor, or the ecdysone receptor. And their pest insects that are of interest are armyworm, budworm, moth, corn borer. So those pest insects are the targeted species. So from this information about the pesticide, we have the protein target, the ecdysone receptor, and then we also have the sensitive species, for example, the budworm. And what we have as knowledge is that there's low toxicity to non-target organisms such as vertebrates, honeybees, and earthworms. But one of the questions was, well, what about beneficial lepidopterans such as other butterflies? And in particular, we were working with our program office partners about a particular threatened and endangered butterfly species. So to generate the results that would be useful in informing the susceptibility prediction, we compared the tobacco budworm ecdysone receptor across the diversity of species. And I don't expect you to be able to read this x-axis. However, it represents the number of different taxonomic groups that were evaluated. This is a typical box plot where we're looking at percent similarity on the y-axis. So comparing the sequence, the primary amino acid sequence of the tobacco budworm to itself, of course, is at 100%. And then within these boxes represents the different species across vertebrates, invertebrates, plants, etc. And this is the susceptibility cutoff that we use for predicting susceptibility across species. To make that brief, it's set by looking at ortholog candidates and functional equivalents. And so from this data, we can see that the ecdysone receptor, as we would expect, is well conserved across invertebrates. And when we look at the ligand binding domain of that nuclear receptor, we see that it's conserved across insecta, merostomata, melcostraca, arachnids, brachiopoda, and other invertebrate species. So like I mentioned to you, we start getting more and more taxonomic resolution as we go through the different levels of the evaluation. So we also were able to identify 10 amino acid residues that were important for the interaction with some of these key pesticides that lead to molt acceleration and therefore death in those pest species. So from our evaluation, again, these are small, but what we can see is there's a list of Lepidoptera species, Hymenoptera, Hemiptera, and then a number of different other taxonomic groups listed below it, including vertebrates. But when we look across these 10 amino acids, we see that across Lepidoptera, they're completely conserved, those amino acids. So these are just the abbreviations for those amino acids on the right-hand side. And then further, when we look at all of the butterfly species for which exist in the data, for which there is existing sequence data, we also see that that's those sequences, those amino acids that are important, are conserved. So if we're bringing this data together, we would say that looking at level 1, level 2, and level 3, the ecdysone receptor is conserved across vertebrates, and specifically if we think about butterflies and considering our endangered species that is also a butterfly, there's consistent line of evidence that all butterfly species that had sequence information available had that ecdysone receptor conserved for that chemical, those chemicals of interest to act upon. And so in the context of the Endangered Species Act, this information can be useful for the determination of may affect and may affect and is likely to adversely affect. So this provides a line of evidence towards chemical susceptibility, specifically looking at the target, and certainly much more information would be brought to bear, but that provides an additional line of evidence. I wanted to give a brief example, but then I also wanted to chat more broadly about efforts in advancing these types of approaches of species extrapolation using bioinformatics in regulation. So as a starting point, we've developed the International Consortium to Advance Cross-Species Extrapolation and Regulation. And our steering committee is made up of the tripartite nature of CTEC, the CTEC organization, so it's the Society of Environmental Toxicology and Chemistry. And we've been working to be able to define the taxonomic domain of applicability, as well as have these communications with decision makers and people that are developing these types of tools, because ultimately we want to define the global regulatory landscape and need for species extrapolation, which would eventually consider both toxicokinetics and toxicodynamics, and bioinformatics would be a small piece of that puzzle. But again, as a starting point, our consortium is focusing on developing a bioinformatics toolbox and thinking about the types of criteria and guidelines that would be necessary for utilization of this type of information for decision making. And then certainly communicating that shared scientific vision. So if you are interested in participating in that international consortium, or at least learning more, feel free to contact myself, or there's a number of us on the steering committee, or Jeff Hodges at Unilever. And then with that, I'll just wrap up with acknowledgements of those working with me at EPA, as well as GDIT, the crew that's the computer wizards that develop the CCAPAS tool with me. And my email is there, so we can certainly follow up if you have further questions, but I guess I'll pass it back to our organizers and we can begin questions.
Okay, well, thank you very much, Dr. Lalone, for that presentation and for those examples and an invitation to collaborate. As a reminder, everyone, please put your questions in the Q&A and we will ask them. And if you can, direct your question to either presenter or both, one or both of them. So it looks like we have a question so far for Michael, going back to Dr. Lowit's talk. And it's a question about the fish acute retrospective analysis that you mentioned. They're wondering if why fathead minnows were left out of the analysis. And is that the case? And can you talk about why?
Sure. The short answer is they were not left out of the analysis. We considered any acute fish study we had, regardless of the test species, that we had confidence in the LC50 value. And so, and I should probably expand upon that a little bit, because I only briefly touched on it, what I mean by that. So there are definitely studies that we have that are, there may be some uncertainty in them, but that they are still suitable for assessing risk. with some level of uncertainty. But for our analysis, we put those in a separate pile because we wanted to have, our question was different, which was really looking at, what was the relative difference in toxicity among the species. So we really wanted to have utmost certainty in the LC50 values we had. So that did not exclude any test species. We actually, I can't remember off the top of my head across the whole data set, but I want to say we had somewhere between 10 and 20 different species in the whole data set. I mean, everything from rainbow trout to, there were some goldfish, and there certainly were fathead minnow. The three that I mentioned just happened to be the most commonly tested within those three groups. So as you noticed, like the cold freshwater species, I mean, that was almost exclusively we just happened to receive rainbow trout studies. With the warm freshwater fish, the bluegill was the most common at 65, but we certainly received many fathead minnow studies as well, just fewer. And so those were included in the analysis, and because of the greater variety of species in the warm water group, we're looking at how that might impact our conclusions. Because what's important for our purposes is not so much the importance of which species, is most sensitive, except in the context of what we normally receive. So when we get a new chemical proposed for registration and the registration data, the data requirements are fulfilled, the chances are we're going to get a very small number of species tested. And that was kind of my point in the slide about the rainbow trout and the bluegill and the sheep's head being the most common. Those are the species we're most likely to get for new chemical testing too. So it's of most interest and utmost importance to understand the relative sensitivity among the species we normally receive for testing.
Okay, that is that's really a lot of really helpful context. Thank you. There's another question for you while I have you. Is there a similar effort to explore the available data and relative sensitivity of endpoints from chronic early life stage for freshwater and marine species?
So let me make sure I understand the question. So we're talking about early life, so the chronic, the ELS studies.
Right.
Is there an effort to compare the freshwater and the saltwater?
Right.
So we do not currently have that effort ongoing. It's a good idea. We do have, incidentally, a different project that's somewhat related, though, that I'll mention that we're working on first, which is to so for most chemicals, we receive early life stage studies for freshwater and freshwater fish and a saltwater fish, which is what the original question was about. However, for some chemicals, also potentially triggered are fish full life cycle studies. So an additional study, which is longer and is carried through long enough to measure reproductive effects. And the question we're asking in that analysis is how often did we actually, and in what situations did we actually get a fish full life cycle study that produced more sensitive endpoints than were already obtained in the early life stage. So in other words, you know, how much value has there been in the past of when those studies have been required of actually conducting and receiving those studies? So that's actually our first attempt at determining what data we really need for the chronic testing with fish. And we might circle back with questions like you asked about, okay, freshwater versus saltwater. Or another line of investigation we're looking at is the potential use of acute to chronic ratios, which gets at potentially having one fish. So maybe testing with the freshwater and then estimating toxicity to the saltwater species. So it's another potential way of getting at the same thing of maybe not needing testing with both species. Good question.
Yeah, actually, there's some sort of follow-ups from a couple of questions starting on that line of, you know, so the original person who asked about fathead minnows said that they're often used in the acute to chronic ratio because the fathead early life stage is shorter and easier to conduct. And then another person is wondering, you know, if there's not alignment in sort of the availability of data or the sensitivity of species, so kind of, I guess, the results of some of these analyses, are there ramifications for the use of acute to chronic ratios? And I'm sure you have that kind of in the back of your mind as you're considering what kinds of analyses to do and what the implications would be. But if you want to talk about that.
Yeah, of course. That's correct. We don't exclusively receive fathead for ELS, but it is very common that we receive the fathead. And although not always understood, whichever species is tested with the ELS, we also require an acute study with that species too. And the point of that isn't so much to estimate toxicity, to be able to calculate an ACR for saltwater species, but to be able to adjust the, do an ACR with the freshwater species. So if the fathead turns out to not be the most sensitive, acutely sensitive species, then we would estimate chronic toxicity. So let's say it happens to be rainbow trout. We would take that ACR of the fathead and estimate toxicity to rainbow trout, chronic toxicity. And then the second question was, so it does get complicated, that there are going to be, there are multiple moving pieces that are going to be going on with any given chemical if we start thinking about reducing the number of tests conducted, whether it's the acute or the chronic, and there is a downstream implication to that. So, and it's going to be complicated. So the way that I'm viewing this is more that we are doing these retrospectives to determine where in the future we can, in a weighted evidence approach, use selectively for each situation different means of kind of getting to the same answer. So in one case, you know, maybe we would be able to reduce testing for the acute but not the chronic, or both, and maybe we would go about it different ways. It just depending on how toxic the chemical is or which species were tested. Or so I guess what I'm trying to say is I don't see that we're going to have like a one size fits all come out of this. I think each situation is going to be a bit of a snowflake, but we want to have the knowledge and the tools to be able to go in with a scalpel basically and make good decisions for each chemical that comes in to say, hey, this is what makes sense for the testing for this specific chemical. So in other words, we still have these basic data requirements, right? We need three acute fish species. We have the ELS on record, the freshwater and the saltwater. But based on what we know for chemical X, here's where we could maybe, we have enough information based on some combination of information. And I just don't foresee at this point, my gut tells me that it's not going to be a one size fits all for all chemicals. It'll basically be, we'll have tools in the toolbox that we can then go and make informed decisions. I don't know if that answers the question, but.
Yeah, absolutely. So it's really good to kind of have that framework of, here's how we're thinking about this. I'm going to ask a couple of questions to Carly now. Dr. Lalone, has CECAPAS been used to look at conservation of toxicokinetic enzymes between species? Although there are lots of, maybe more context. Although there are lots of TK pathway species combos, it seems like a fruitful area to understand species sensitivities to pesticides, et cetera, on a broader basis.
Absolutely, and great question. So if we understand, say, metabolizing enzymes that are critical for the action of a chemical and the metabolism to the active ingredient, for example, we have done a number of different examples looking at a variety of cytochrome P450s, but that does get challenging, particularly in the realm of pesticides. The sequence availability for invertebrates decreases significantly, of course, from model organisms as well as from vertebrates in general. But like I had mentioned, there are consortiums that are working on generating more whole genome data for multiple different types of species, including invertebrates, aquatic invertebrates, marine species, for example. So where there are these sparse numbers of species represented for sequence data, because of sequencing technologies growing so rapidly and improving so rapidly, there's great promise to bring in more sequence data. And then the other challenge on top of that, of course, is quality of annotation. And when we start looking into more invertebrate data, a lot of times, we just have to pay close attention to what the annotations are and what the sequences are, and how well they're represented. So that being said, we have done case examples looking at a variety of cytochrome P450s in bee species, but also, with some of our pharmaceutical case examples, we've looked at a variety of different SIPs as well, and in this context. And then I had also mentioned kind of the bioaccumulation potential of PFAS. So thinking about those proteins important for bioaccumulation is another means to utilize the CECAPAS tool and think about, for example, liver fatty acid binding protein, and how we might predict bioaccumulation potential. So there's multiple different applications of the tool. I really focused on kind of that chemical susceptibility and knowing the target. But the metabolizing enzyme, if that is known, that would also be the target that you would begin a CECAPAS query. So great question, and yes, that is an application of the CECAPAS tool.
Great. Another question asks, and maybe this kind of coalesces with a question I had jotted down, what about the CASER activities. And so it's asking how you plan to build confidence around using these tools for regulatory decision making, and maybe that's an opportunity to talk about that collaboration.
Absolutely. And I think that bringing together the decision makers that have different pipelines that they're using in their decision making processes and the types of data needs that they have, and bringing together the developers of these tools, and not just CECAPAS, but certainly there's many others that compare using bioinformatics, and certainly there's the toxicokinetic aspect as well. But bringing these groups of people together to discuss the needs and the criteria, what gets these types of data into a pipeline for decision making. So not just developing our tools and our silos within our individual organizations, but working across global partners and thinking about developing examples and bringing in the decision makers to help organize and understand what the needs are. Those are the most critical, I think, interactions that can move this type of science into regulatory decision making and into the hands of those that would benefit from it most, I think, as far as particularly considering surrogacy issues. And so in building confidence, I think we need to identify the champions like within organizations that are really willing to begin to understand how they could apply these methods. For example, you know, we work with some really, I think, visionary program office partners that, you know, reach out and they want to apply these types of approaches to their decision-making processes. And so I think bringing that in on a global scale and having those interactions with others would only improve our ability to utilize the tools in a manner that would be useful for multiple organizations, and bring to like Michael was just saying, it's not going to end up being one tool that solves, you know, many different issues, but bringing together the right data that can help bring in different lines of evidence or weight of evidence for understanding chemical effects across species. And that's really what this consortium aims to do, is bring people together that can work together on those different issues.
Great, thank you. A quick question for you about the tebuphenozoid, the work in butterflies. Is that published?
It is, yep. So it's in a 2016 Lalone et al. Paper on CECAPAS, and that was one of the case examples described.
Great. And just to turn everyone's attention to the screen on your, the slide on your screen, because we had a question about whether the presentations will be available to participants, which yes, that is the case. So the recording and the slides will be posted at the website on your screen right there, where you can also see actually all 9 previous webinars in case you'd like to catch up. Okay, thanks again, Carly. We have another question for Michael. The person is wondering, do you think marine versus freshwater species sensitivity differences are due more to exposure considerations rather than actual sensitivity?
Not certain. Maybe they can, I think I know what they're getting at, but I'm not 100% sure. I mean, so I'll just answer and then they can always follow up. So, I think they're asking more in the field, maybe, but maybe actually, can we just ask?
Yeah, I don't know if they're still on, but we can
Well,
chat.
I mean, the short answer is in the field, I'm not sure, but in the lab, I mean, it's purely, you know, these tests are standardized. They're following standardized test procedures. So really the difference between the saltwater test and the freshwater test is primarily, other than the species tested, you know, the big thing is the salinity. Yeah, I mean, the differences that we're seeing in those tests should be primarily due to the test species.
Okay. I do see
But yeah.
the person who asked the question, Maria Blanco, is on the line. I'm gonna try to unmute them and see if they're available. Let's see what happens here. Maria, you are unmuted on our end. If you can go ahead and.
Hi, I think I'm on mute now. Yeah,
Yeah.
it was just, can you hear me?
Yes,
Yep.
we can.
Okay, good. Thank you, Michael, for the nice presentation. I was just curious about the results, saying that species or the salt species, let's say, were less sensitive than the fresh water. So we were wondering if these could be more like a reason because of the exposure of the experiment rather than the species themselves.
Right. Yeah, I think because we're using standardized test designs, unless the salt, unless the fact that you've got a more saline solution is in itself causing some impact on the toxicity of the chemical, relative to it being in a freshwater media. Other than that possibility, I would expect that the results that we are seeing are primarily due to the sensitivity of the tested species. And that also doesn't mean that our results, I also want to stress, our results don't mean that, like, on the whole, like across all saltwater species and all freshwater species, that freshwater species tend to be more sensitive on the whole, or that if you put these specific species out in the environment that this is the result we would get. This is very specific to the testing of these species under test conditions. And the reason that's important is because that's what we use as the baseline for assessing risk. So the idea is that if, the freshwater, regardless of the reason, if the freshwater species tend to give us that more sensitive value more often than the saltwater species, then that saltwater species, except in a select number of cases, isn't giving us any value added to our risk assessment. And the reason for that is that our screening level risk assessments, we use the most sensitive species tested to, it's the baseline for our risk. Where it could come into play, for a more refined risk assessment is when we start, kind of like what Carly was getting at, when there's more of a need to look at a species sensitivity distribution, then it becomes a little bit more important. But then, of course, we're only basing it on 3 species too. We're only looking at the lab data. But so although I think it's a really good question and it's an important question ecologically, but from within the boundaries of our purpose of why we're looking at this and thinking about in the context of those studies, on some level, it almost doesn't even matter.
Okay, thank you.
Yep.
Maria. Okay, well, we have one other question for you, Michael, but I think you may have already sort of addressed this. It's just asking what are the next steps in your acute fish work? So what are the implications? And you've kind of talked broadly about that already, but just to see if there's anything you wanted to add.
Yeah, I can just reiterate. I mean, we're finishing up the fine details of the analysis. The intent is to publish those results in the open literature, and then as and this may be the part that I could expand on a little bit. So as a completely independent and separate process, we will internally take those results and determine if there's conditions under which we can grant or that we can potentially grant waivers for future submissions. And then if so, under what conditions, which chemicals do we feel the most comfortable with? It won't be all of them. And if we, say, okay, we'll waive study X, do we have to make any assumptions in the absence of that? And here's what those are. That'll be a separate process because that will be more about policy. It will be guided by the science, but it'll be a policy decision. And so what that could look like is we would put out potentially a waiver guidance that would explain all those scenarios. And we've done that in the past. So for instance, some of you may or may not be familiar, we've already done a retrospective analyses with the acute avian studies, the oral and the dietary. And we have waiver, so it was the same sort of situation. We did an analysis, the analysis was published, and then a separate guidance document was created describing under the conditions where we would consider waiver requests for the acute dietary study in this case. So that's kind of the process. So it's a two-step process.
Great. That's really helpful. Well, that's the end of our Q&A. I want to thank all of you on the line for listening. I especially want to thank the speakers, Dr. Lalone and Dr. Lowit, for sharing your time with us, sharing your work and expertise. And to my co-hosts, Gino and Bridget and Amy, for helping to put this webinar on. Again, as we mentioned, the slides and recording will be available on the Science Consortium website shortly. And feel free to get in touch with any of the organizers or the speakers if you have more questions or you want to get involved in any of the activities that they mentioned. With that, I guess we will close the webinar. Thanks very much again to both of you presenters and to all of you for your questions.
Thank you.
Thank you.
Yep, thanks. Bye.